So as you said, we work in the Translational and Synthetic Biology lab at the University Pompeu Fabra in Barcelona, that is led by Marc Güell. And there in the lab, we have two main research lines. We do engineering of the mammalian genomes, so cells that are present in the human body. And we also do engineering of the skin microbiome, so the bacteria that live within our skin. And in both cases, we modify the genes, or we introduce new genes in either the cells of the humans or these bacteria that lives in our skin with a therapeutic purpose. So today we will talk about this mammalian genome editing research line and the things that we do in the lab, and now we are trying to apply to treat this disease. So I think I don't need to do much more introduction about this disease and the cause of this disease. But what we really like about this model is that we can treat the mouse model either with direct editing of the cells in the patient, or the mouse model in this case, and also ex vivo, because this laminin, being an extracellular protein, it can be expressed not only by the fiber but by other cells that will then produce the protein, and it will be in the correct place. So in our lab, we work with a mouse model for the laminin deficient disease, and it's this DY2J model. It's a mild phenotype. It's not the most extreme and affected mice that right after birth you can see the phenotype. In these mice, the phenotype arrived upon 6 to 8 weeks after birth, and then they can live up to 6 months. And the life expectancy of the mice is 2 years. So this is a good model that recapitulates well the disease in the patient. And we wanted to do this ex vivo treatment of the mouse model because we saw this publication that says that in inflamed muscle, there is circulating cells that can infiltrate into that muscle. So if we modify the cells that are in the bloodstream, and then these cells can reach the muscle, then we can express the laminin in these cells in the bloodstream that then reach the target tissue. And then we wanted to do the same thing that they did in the publication, and we took bone marrow cells from the bones in the mouse. Then we modified them with lentiviral vectors to express green fluorescent protein, and we put them back in mouse that were conditioned, and then we caused an acute inflammation in their muscles. And we saw that not only in the peripheral blood we could see these modified cells, but also in the cells that were infiltrated in the muscle, we could see these cells expressing the green fluorescent protein. So we also see this publication that they took bone marrow from healthy donors and they put them in affected mice, and they could see a recovery in the life expectancy and also in the strength of the muscle. And we did the same thing. We now took bone marrow cells from wild-type mouse, modified them with DFP to be able to track them, and then we put them back, in this case, in affected mice. And we could see the same results that they published, and we saw an increase in the life expectancy of these mice. So then we wonder, what if we take the cells from the affected mice and then modify them to now express LAMA, because they have a mutation in the LAMA gene. So now these same cells that could not express LAMA, we modified them to express LAMA and put them back in the mice. And this is what we did, also with lentiviral vectors. And then we treat the cells outside of the mouse, and then we put them back. And we could see even a better rescue in the survival. So these mice lived until 2 years, and we saw that when we when we didn't treat them, they start dying after 20 weeks. And by 1 year of age, all the untreated group died. But the treated mice survive as much as the wild types. We also see a recovery in the strength of the muscle. It's not as the wild type, so they were still affected, but they could move better and they have greater strength in the muscles after 12 weeks of treatment. And that's all that I wanted to share with you today. This is our group at UPF Barcelona, and I also want to thank the Patient Association here in Spain, ImpulsaT, and our collaborators and advisors, both at our institution and in Vall-d'Hebron Hospital. And I will be happy to answer any questions.
Thank you so much for your presentation. I'm in the meantime looking in the chat, and it has been quite silent so far, Avencia. So I don't have any questions yet. Are there any questions remaining? Nobody has to be shy about it. I still don't see anyone. You could also showcase your else. I think we're going to introduce something else then, at least to get some interaction going on. At the bottom, there are a couple of buttons, and one of them says reactions. And you can give every speaker a warm applause. Let's try doing that, and at least we see that people are actively participating, because thank you so much for your presentation. I see a couple of questions coming in. Once again, the same question we had before. Is this a one-time treatment, or would this be an ongoing treatment?
So we expect this to be a one-time treatment, because then once the cells that are modified engraft into the bone marrow of the patient, they will continue to express Lama their whole life.
Okay. And the second question is, can it already be tested on humans, or is it already in that phase?
It's not already in that phase. So the problem with these lentiviral vectors that we tested is that to produce such a big gene and to encapsulate such a big gene, then the efficiency of the production drops, and then it makes it challenging to produce enough virus to treat into a patient scale. So we are working into modifying the way that we introduce Lama in these cells that come from the bone marrow. So I think there is still some work to do for translating this into the patients.
There's also a question from Helene Stelwa. Given this method works, does this therapy only stop the muscle deterioration, or is there a chance for building up new non-affected muscle fibers?
So that's a really good question, and we are still investigating why we see the recovery and the survival and in the muscle strength. So we are running a few tests on the mice to see whether or not there is an increase in laminin expression and dislocates where it has to be, and we see the fibers with the architecture that is expected in a healthy patient. So we are still not sure why the therapeutic effect is seen, and we are looking into that.
And for this treatment, do you also need immune modulating medication for it to work?
So not in the mice, and since it's an autologous transplantation is the same patients from the same cells from the patient, we don't expect that to be the case, but we will have to see if there is any reaction upon the laminin alpha expression, for instance.